A few people on the call. My name is Chinakaho Melissa. I am a training coordinator with Ilwazi Open Data Science Platform. Today we have with us Dr. Melissa Nel, and she will be presenting on exploring the application of pangenome reference graphs to rare disease diagnosis. To introduce Dr. Nel, she has completed her MB-CHB in 2008 and also her PhD in 2016 with the University of Cape Town. She is currently a senior lecturer in the Department of Medicine at University of Cape Town, and she's working directly with the Neuroscience Institute, where she leads a neurogenomics lab. Currently, Dr. Nel is collaborating with the UCT Neurology Research Group and the ALS Africa Network to study the genetic basis of amyotrophic lateral sclerosis and other inherited neuromuscular disorders in African populations. She is passionate about African-led genomics research and local bioinformatics capacity building to drive the real-world implementation of genomic medicine in resource-limited settings. Her neurogenomics lab seeks to partner with DNA sequencing service providers in South Africa to enable her team to deliver cost-effective and rapid analysis of genomic data for rare disease diagnosis according to international best practice guidelines. Her group aims to provide ALS and NMD patients access to genetic diagnosis, opportunities to contribute to scientific discoveries through participation in global research, and to promote their equitable participation in therapy development and trials. I'll hand over to you, Melissa, to give your presentation. Thank you.
Thanks very much for the introduction. Unfortunately, I'm having an issue with my presentation freezing at the moment. Sorry about that. Do you see the second slide at the moment?
You're not sharing screen at the moment, so maybe try sharing.
Okay, I'm just going to force quit and try and open it again.
Okay.
Sorry about
Alternatively,
this.
okay, no worries. Or you can email me the presentation and I can go through the slides for you while you present if this doesn't work out.
Okay. Okay, I've opened it again. Let me try and share it now. How does that look?
It's perfect. Thank you.
Okay, great. Thanks once again for the introduction. So the title of my talk today is Exploring the Application of Pangenome Reference Graphs to Rare Disease Diagnosis. And I would hope that I can share some of our preliminary experience in my lab with working with these graphs in our clinical research. So the focus area of our research is neuromuscular disorders, abbreviated as NMDs. And these are a heterogeneous group of disorders which are characterized by pathology of muscle, neuromuscular junction, nerves, neurons in the spinal cord, as well as brain. And the clinical presentation and age of onset of these disorders varies. A common feature across all of them, however, is a genetic origin based on Mendelian gene mutations. And in fact, most neurological disorders resulting from genetic mutations are neuromuscular disorders. So neuromuscular disorders have a very diverse genetic origin, meaning that very many different genes are involved in their presentation. So I've just shown you three representative pie charts from some of the major categories, being genetic neuropathies, congenital myopathies, and limb-girdle muscular dystrophies. And as you can see, there is definitely a vast array of different genes that can cause these disorders, some being more common than others. Secondly, in addition to there being very many genes involved, within each of these genes, there are very many different pathogenic variants in each patient. And also, there are many different kinds of variants that can give rise to these disorders. So this schematic just shows you the examples of single nucleotide polymorphisms, small deletions and insertions, and then the class of variation, which I've highlighted here, which is referred to as repeat variation, or as we refer to them, short tandem repeats, is very relevant for us in the field of neurology, which I will show you in the next few slides. So these short tandem repeats, or STRs, are DNA sequences with a repeating motif of 1 to 6 base pairs, and they contribute to the genetic pathogenesis of various neurological diseases. As shown in the schematic on the right, you can see a sort of general schematic of a gene, showing you that these repeat expansions can occur both in coding and non-coding regions. And then the nucleotide shown in brackets is the motif that becomes, that is basically repeated to give rise to these short tandem repeats. And you can see that they are associated with disorders such as amyotrophic lateral sclerosis, or ALS, as well as neuropathies and ataxias. So a very diverse array of diseases. So we call them pathogenic repeat expansions when they have expanded beyond a certain disease-causing threshold. And this is just to show you 2 examples. So you may have heard of the C9orf72 gene, but this is a gene that harbors a repeat expansion that causes amyotrophic lateral sclerosis. So if you look at the repeat distribution shown here on the left, you will see that this is just a normal population, that most individuals have repeat motifs of between 1 and 12 units, whereas those that cause disease are more than 30 units. So the profiling of these STRs is traditionally done using wet lab techniques such as repeat prime PCR. But increasingly, there have been the development of computational tools to actually genotype these repeats, these short tandem repeats, using whole genome sequencing data and even whole exome sequencing data in cases where the repeat expansion is close to a coding region. So in this slide, I just wanted to give an overview of the broad genetic testing strategy that we employ for these neuromuscular disorders. And the slides leading up to this showing you that there are very many genes involved and very many different kinds of variants, explains why whole exome or whole genome sequencing is the method of choice, because in any given patient, based on the phenotype, so the features that the clinician observes when examining the patient, it can be very difficult to hone in on a particular candidate gene or even group of genes that might be causing the phenotype. And so it becomes very useful to be able to screen many, many genes. And most of the analysis that we do in the lab is for whole exome sequencing data, although whole genome sequencing data would be preferable. We really just rely on exome sequencing because of the cheaper cost for this sequencing technology. So here we're able to broadly screen all the protein coding genes and look at short variants, including short insertions and deletions. And then in some patients, we have whole genome sequencing data where we're able to do the same, but also start to explore these repeat expansions, as well as larger copy number variants or structural variants. So overall, the diagnostic yield across this group of neuromuscular disorders is around 50%. And I just wanted to highlight that in this example pie diagram shown here for patients with limb girdle weakness. So if we employ a combination of single gene testing in some cases, gene panel assays, and then short read exome and genome sequencing, including profiling of these STR expansions that I showed you, as well as other analyses to try look at copy number variants and structural variants, the best we really can hope for is a solve rate of around 50%. So there is this remaining proportion of patients, approximately half of them, in whom we are not able to identify a causative genetic variant. So this pie diagram is obviously of published data, but our experience examining exomes and genomes from South African patients with this group of disorders has been very similar, in that we also are only able to solve around 50% of the cases. So in this slide, I wanted to map out our sort of current thoughts around the missing heritability. We speak about this concept of missing heritability because we believe that this group of disorders is heritable. So the fact that we expect that there is a genetic determinant in all the probands that we examine. And so in those in whom we don't find an answer, we assume that we are missing the causative genetic variant. So in the gray boxes here, I've tried to unpack where we and others think this missing heritability might arise from. So the first aspect is knowledge gaps. So for example, in the variants that we call in the VCF files that we generate from this NGS data, the variants are there, but it could be that we just don't know their true biological relevance or meaning. For example, there could be novel disease genes harboring variation that we simply don't know causes neuromuscular disorders, and so we overlook these in any individual patient analysis. Many of these causative mutations are just missense variants, and obviously every individual has a number of deleterious alleles, and so it can be quite tricky to sometimes figure out which ones might be disease causing. Then the second aspect that's been considered to explain this missing heritability is around the reference genome and specifically reference genome bias. So the question sort of researchers have been asking is whether the reference genome that we use, for example, current linear reference genome HG38 or the more complete CHM13, whether we are missing any variants in perhaps some of the newer regions in CHM13 that we haven't evaluated before. And the second issue related to this is whether the reference genome is biased in terms of the representation of diverse haplotypes. And perhaps if we had more representative reference genomes for the populations we were analyzing, we might recover causative pathogenic variants. And then lastly, perhaps the missing heritability is explained by the sequencing technology that we're using. So to date, this has largely been short read technology. And we know that we have very limited ability to see the full extent of structural variation in genomes using the short read technology. So perhaps that is why we are not able to uncover the remaining pathogenic variants. So for the next part of this talk, I would like to share with you our preliminary work where we've looked at reanalyzing our existing short read data on patients with neuromuscular diseases for SNPs and indels using pangenome reference graphs. So before I get to that part, I just wanted to reflect briefly on the current HG38 linear reference genome, which is what we were using up until recently in our analysis pipelines. So we know that this genome has long stretches of DNA, which are in fact highly specific to one individual. And we know that reads with non-reference alleles will have lower mapping rates to this genome. So although we know it's problematic, for a number of reasons, change is also very tricky because all of our annotations, and particularly those for clinical genome interpretation, are built around this reference genome. So it becomes quite challenging to think of moving to another option. However, the human pangenome reference is now available, and this includes 47 phased diploid assemblies from a cohort of genetically diverse individuals, and should be a far better representation of global genetic diversity rather than the single linear reference genome that we're used to working with. So before I go on to the pangenome approach that we have used, I wanted to touch briefly on just some aspects of the ancestry around our patient samples that we analyze. So in this schematic, I am showing you the global ancestry of a subset of patients with neuromuscular diseases in whom we've identified a pathogenic variant, where each individual is represented by one of these columns, and the colors are then corresponding to the contributing source populations that are present in each individual. So although we have some individuals of, for example, European ancestry or African ancestry, a large proportion of the patient cohorts, I would say roughly half, are individuals of admixed ancestry, where we typically have 5 contributing source populations as shown in the different colors in every individual. So a very diverse patient population group, including sort of a noteworthy mention is that African genetic ancestry component in many of these individuals has a prominent Khoisan origin, which is very much understudied and underrepresented. So then if we move to the local ancestry, as I said, we do have some patients of European ancestry, those of African ancestry, but then those who are admixed have multiple ancestries represented. And this, is different in basically they have different local ancestry tracts along their chromosomes. So different regions have a different ancestry origin. And so the challenge here would be that in order to, develop a suitable reference genome for the analysis of an admixed population, we really need multiple reference populations to be represented in that reference. So then moving on to the personalized pangenome reference approach, which is the pipeline that we have adopted and tested in our lab. So this is a new approach which was inspired by local ancestry methods. So it was very relevant, seemed a very good fit for our analysis questions and our population sample. And this approach samples haplotypes that are similar to the sequence genome according to KMA counts in the reads. And the rationale behind this is that, as I've stated in the last quote here, having the right variants in the graph has a greater effect on mapping accuracy than having variants not present in the sequence genome. And so basically what happens here is instead of using the entire HPRC pangenome reference, one is able to extract haplotypes that, best represent the sample that you're analyzing, and in that way create a personalized pangenome for the sample, which should improve accuracy. So for the NMD scan analysis pipeline that we use, I'm just showing the first part of it, which is where we use, obviously we have to map our sample reads to a reference in order to do all the downstream analyses. So we have used the CHM13 referenced graph, where we basically are using the personal pangenome pipeline to map sample reads to a personal pangenome graph, which is extracted from the main graph. And so we're getting the benefits of using the new CHM13 reference, as well as the benefits of including greater haplotype diversity in the reference. And one very important aspect of this pipeline is the surjection step. So this enables us to project the graph alignments onto a linear reference, which maintains compatibility with our downstream tools and clinical diagnostic workflow, particularly all the annotations. So as you see here, although we are using the CHM reference graph, we are subjecting it back to a HG38 BAM to maintain this compatibility. And obviously it would, I guess, be preferential to use CHM13, but there are a number of limitations in what we can actually do with the data mapped to that reference genome. So this is just to show you some of the results just from one representative patient sample, which was whole exome sequencing data, where we compared linear alignment to the HG38 reference versus a personal pangenome alignment to the CHM13 graph, and then subjected back onto HG38 to do variant calling using deep variant. And as you can see, although there was a large overlap between the call sets for both SNPs and indels, we identified novel variants, which we did not identify, using the graph, personal pangenome graph method, that were not identified using the linear alignment. And some of the variants which we identified in the linear alignment approach were not identified on the graph. So just to speak a little bit more about this in detail. So with respect to those variants that were only identified on the graph, we would refer to these as novel variants. But in fact, when we reviewed all our cases we did not solve any additional cases with any of these novel variants, meaning that none of them were pathogenic variants in known NMD genes. And similarly for the false, well, not similarly, but just to comment on what we would refer to here as the possible false variants. Obviously, we can't say that for sure, and we haven't, under the challenges here, we haven't spent an extensive amount of time doing internal benchmarking around the differences between these two call sets and what they really represent. But at least at this point, we can see that we do get similar results to other benchmarking exercises, which have shown a greater recovery of rare variants using a linear, using a pangenome graph reference as opposed to a linear reference. Although the clinical relevance of these novel variants remains to be established in general for rare disease diagnosis or for potentially enhancing rare disease diagnosis. So a couple of aspects which we did find in our comparison. So we found that alignment to the CHM13 reference graph resolved incorrect gene representations. So we know that there are genes which are falsely duplicated in HE38 compared to CHM13, as well as genes which are falsely collapsed between these two reference genomes. And a subset of these genes are known to be medically relevant for inherited diseases, although none specifically for the neuromuscular disorders that we are interested in examining. But it was interesting to examine these regions between our linear versus graph alignment and to see whether we could improve on the analysis of these regions. And indeed, when we looked at the CBS gene, which is one gene that is shown to be falsely duplicated in HG38, if you compare, this is the same sample aligned using these two different methods. So you can see in the linear HG38, you have these reads with a low mapping quality. And when you use the CHM13 graph, pangenome graph for alignment, you see that the mapping quality improves because you now have a single gene representation rather than a multi-gene representation, which leads to the multi-mapping problem, which you see in the first panel. The same was also true for the other falsely duplicated genes that we examined. So again, we saw that this problem was resolved when using the personal pangenome pipeline. Although, as I mentioned earlier, these particular genes are not clinically relevant for neuromuscular disorders specifically, although they may be relevant for other rare diseases. Also, genes that were falsely collapsed in HG38 compared to CHM13, we also saw that the representation of these genes differed between the graph versus the linear alignment. And we were able to detect some coverage differences. Obviously, the coverage being reduced when the genes did not have the collapsed representation. So the other thing we were interested in looking at were these so-called dark regions of the genome. So these are regions that have been shown to be basically poorly profiled using short read technology. And this is just showing you a word cloud of a sort of collection of medically relevant genes, many of which are involved in neuromuscular disorders, for example, the nebulin gene and the selenon gene. And so we were interested in having a look at these genes and seeing whether the personal pangenome pipeline could improve our ability to screen these genes. So before I go into the results, I just wanted to explain there's two main categories of these dark regions, which is very well explained in this paper by Everett et al. From 2019. But these genes are described to be either dark by depth or dark by mapping quality. So those that are dark by depth are variable between samples. The example shown here is for the HLA-DRB5 gene. And these genes have got a low number of reads. And this is due to library preparation differences, high GC content, or else high sequence polymorphism, where highly polymorphic reads originating from samples do not adequately map back to reference genomes. So this would be the dark by depth category. And then another category are genes which are dark by mapping quality. So these arise from duplicated genomic regions, so where your aligner has difficulty placing them in a unique location. And so effectively, these genes are lost to analysis due to reads with a poor mapping quality. So when we looked at some of these examples and compared them between the linear and the graph alignment approaches, for example, the highly polymorphic HLA-DRB5 gene, which is dark by depth, we saw that a similar mapping quality, but there were definite coverage differences with improved coverage of the region in the graph compared to the linear reference genome. And then when we looked at some of the genes which were dark by mapping quality, so for example, the HSP A1A, A1B gene pair, which are highly similar genes, we could see that although there's an apparent difference in the linear versus the graph, we see that the improvement in mapping quality is really negligible. And so using the graph does not offer any advantages in examining these highly homologous regions of the genome. And again, the same was true, just to show you another example for the SMN1, SMN2 genes, which are also dark by mapping quality. Again, we see the same picture. There's really no improvement in mapping quality that would translate into any differences in variant calling. And this makes sense because these genes, SMN1 and SMN2, are large and they are highly similar, only different by a few base pairs in certain positions. And so this is really a limitation of short read sequencing technology, and there isn't any improvements that one can gain by using a different reference genome to resolve these regions. We also looked at STR genotyping. So as I mentioned, this is a really relevant class of genetic variants that we need to examine in these patient genomes. And across all the loci that we looked at, we did not see any differences in the linear versus the personal pangenome graph alignments. So we had identical read coverage and identical genotyping results. So there was no improvement that was offered for this particular category of variants. So to summarize these findings, which would be our reanalysis of short read exome and genome sequencing data, we found that the personal pangenome graph alignment and variant calling, although we didn't do the benchmarking ourselves and look into this in a lot of detail, we feel that it should be more accurate based on published results. Importantly, we found the pipeline to be feasible in that it was simple to run. It had a single sample analysis mode, which is important for us because we rather than analyzing data in batches, we analyze the data on individual patient samples as they come in. We found it to be fast, and it was compatible with annotation sources and downstream tools on account of the HG38 surjection. Despite the increased coverage, there was no clear benefit for well-characterized NMD genes, and we didn't find any advantage for profiling of disease-associated STRs. We compared all our data to date using, obviously this was all done on a linear alignment, and we repeated all of the samples using the personal pangenome pipeline and recovered the same pathogenic variants. So it was equal to our previous work. And we have decided to, based on the above, adopt the personal pangenome analysis route as the standard analysis pipeline. At the moment, we've only focused on short variants, and we're interested in looking at structural variants in future. And so it will be interesting to see whether there are any differences for this class of genetic variants. Although having said all of the above, we know that short reads remain a limitation, and not one that can be overcome by using different reference genomes. And so that is something that we acknowledge. And unfortunately, it just remains prohibitively expensive to use long read technology on every patient as a first-tier test. So back to the slide of mapping out the missing heritability. We've done some work, as I've shown you, remapping our short data to more representative reference genomes in a graph structure. But we really feel that the next frontier of novel variant and disease gene discovery is going to be uncovered by long read sequencing, which will be able to uncover structural variants that may underlie this group of disorders and perhaps for rare diseases more generally. So our future work in how we propose to take this approach forward. So our first tier analysis will remain any combination of single gene testing, short read exome sequencing, and short read genome sequencing with structural variant analysis on patients with neuromuscular disorders, simply from a cost perspective. As I said, it would be prohibitively expensive to use a long read on every patient, and it would also be completely unnecessary, since we know that a large proportion of the cases which we solve harbor pathogenic missense variants, which are very easily detectable by exome sequencing. So we will continue to use this as a first tier approach, using the CHM13 graph subjected to H238 to allow us to do the relevant annotation and filtering. And then for those patients which we are not able to solve using the first tier analysis, we will move to second tier analysis, where we will generate PacBioRevio long read whole genome sequencing on these cases. We'll then adopt a stepwise approach to the analysis of this data, firstly aligning it to the linear H238 reference genome, and doing the analysis and seeing whether we can detect any pathogenic variants. And if that still remains negative, then we will pursue alternative strategies, such as aligning the long reads to a pangenome graph, and even exploring the possibility of doing personal assemblies for individual patients. So I wanted to end by just sharing a clinical conundrum that we've uncovered using short read sequencing data, because it speaks to our move towards long read and also possible other applications of pangenomics. So we have, over the course of our sequencing efforts, identified 4 African ancestry patients with a neuropathy phenotype. And this has been on account of identifying heterozygous, 2 different heterozygous variants in a gene called MPV17, which would be a new disease gene assertion for this particular phenotype. And importantly, in order for this to be real, we would be pursuing an autosomal recessive inheritance picture in each of these probands. So what that means is we would want, in order for these to be proven to be pathogenic, we would need to demonstrate that each of the variants is on different alleles. So showing you these 2 configurations here, we can either have the variants in cis on the same allele, or we can have them in trans on different alleles, meaning that both copies of your MPV17 gene would be impacted. Now, because we detected these variants using short read technology, we were not able to phase them. And so the configuration, meaning either cis or trans, we were not able to determine using the short read data, but it remained a very compelling possibility that these were in fact a novel cause of Charcot-Marie tooth disease in African ancestry populations. And so we wanted to pursue this further in order to try and establish the configuration of these variants. And so we moved to targeted long read sequencing. So effectively, we designed primers based on the linear H238 reference genome to amplify the MPV17 gene. So the certain exons that were harboring these 2 variants, in order to try and effectively phase them and determine which configuration was present in each of these patients. And I'm just showing you one example here. We did this for all 4 of the affected probands, and they all look the same. And it was quite surprising that instead of giving us a clear answer, we picked up a mixed picture, which I show you here in these 2 black boxes. So we detected the variants both in cis and in trans, so both together with one another on the same read, as that was in 50%, and then the other 25% of reads had only one of the variants. And so this was really interesting, because we then questioned whether there perhaps multiple gene copy numbers of this gene in in these patients or maybe in African ancestry individuals in general, whether there's perhaps a pseudogene or some paralogous gene region. And in fact, when we amplified this region, we effectively had a mixed population. And so it's been very challenging to try and actually decipher what is going on within this gene within these patients. So this just leads me into the last slide, which is not something that we've delved into in any great detail, but just to show you some preliminary work utilizing pangenome graph visualization, which one of our students has begun to investigate for these two highly homologous genes, the SMN1 and SMN2 genes. And she's looked at the HBRC graph and looked at the copy numbers of these genes and their orientation within the graph. And so this just speaks quite nicely to this clinical conundrum that I presented, where we are interested to know whether there are architectural differences in this region or copy numbers or different configurations that perhaps we're not aware of. And so being able to visualize the pangenome graph structure of a representative population could be a very useful avenue as we try to dissect the causative variants in these population groups. So that's really all the results I have to show. It's a new field and one that we are still exploring, but overall we think it holds great promise for our research, particularly given that our patient population is so understudied and is the possibility of them harboring a novel genetic variants is quite high. So we look forward to collaborating and working and learning from the RefGraph project to try forward in the future. So to end off with, I'd just like to thank everybody in my lab, all my collaborators, as well as the funders for supporting this work, and thank you for your time.
Thank you, Melissa, for that presentation. It was quite informative, and I'm sure the people on this call will have questions, as we do have individuals who are working on the pangenome and RefGraph project present in the meeting. So I'm gonna open the floor for any questions or comments that the audience might have for Melissa. You can raise your hand or type in the chat box to ask your question. Chris, you can go ahead.
Yeah, I just had a quick question, because it seems like the direction that your research is going is very much going in the direction of long reads. And so do you see a future for short reads in this field still, or do you think that eventually everything is just because of the cost, or is it just going to be going in the direction of long reads no matter what?
I can't see, do you still hear me?
Yes.
Yes. Yeah. So Chris, I think, yeah, that is an interesting question. And actually our original workflow was to first, it's kind of evolved over time as, new sequencing technologies become available and the cost. But typically, if the clinicians have a strong clinical suspicion of a particular phenotype where there's a particular gene that's quite common, there's still a place for doing a single gene test for that gene first, just to rule it out. But then our current thought is that the best approach is to do a short read exome, because that has come down in price a lot and is now accessible to us in South Africa as well, and that will solve around 50% of the cases, with a relatively simple variant in the exonic region. But then before we would typically think of moving to a short read whole genome, and now we're more in favor of moving straight to a long read whole genome if an exome is negative. I think the price of a long read would have to come down significantly for us to warrant that as a first line diagnostic test.
Yeah, we've been a little lucky here because we have access to the newer the Revio, and so the cost is now 1/3 the cost that it used to be, because you get 3 times more data. But the direction that I've been seeing is that people are doing kind of a 5x 10x coverage, so they could do some basic variant calling but not assembly to keep the costs somewhat low. But yeah, I think that even with that little bit of coverage, you can do structural variants, which I found is kind of interesting. You can actually get quite a bit of resolution even at low coverage. So, all right, thank you.
Hi, Melissa, this is Yasmina. Thank you for the talk.
Hi, Yasmina.
I just wanted to ask, to what extent has the diagnostics of neurodegenerative diseases in the clinics, to what extent are you using genetic testing? I mean, if it's for one gene or several genes, are you doing it like on a routine basis or?
No, so the single gene testing, some of the genes are available through the NHLS, which is our National Health Laboratory Service. And so the clinicians that we work with are based at Groote Hospital, which is a tertiary referral hospital here in Cape Town. So they would be able to request for public patients these single gene tests through the NHLS. But this the diagnosis, although it's standard of care, in other places it's not standard of care in South Africa, and so that these patients are only accessing diagnostic exome and genome sequencing as part of our research project. So it's not available on request to clinicians, if that answers your question.
Yeah, thank you.
Melissa, I see there's a question from Paul in the chat box. It says, thank you, Dr. Melissa, for the informative presentation. Other than the variant counts, is there another way of determining significance of pangenome reference versus linear references?
Yeah, so I think that also depends on your application to a certain extent. I mean, my perspective is very much rare disease focused and on profiling the variation in particular genes. But of course, the caveat is that we only know what we know so far about the disorder. So I'm not sure that we necessarily know the relevance of these additional variants and what they actually mean. Are they in biologically relevant regions of the genome? What's the impact? I think that's still quite difficult to establish, but I think we've assured ourselves that this approach can't be, we can only be doing better than what we can do on using a linear reference. So we may not know the full extent of the advantage or what exactly it's, this information means, but I think I'm quite comfortable that it's definitely equal to and better than using a single linear reference.
Thanks, Melissa. I think we also have a hand raised from Sean.
Yeah, thanks, Sinikau. Thanks, Melissa. Thanks for the talk. It was great. I was curious. So the study that you did, you were using the HPRC graph, right?
Yes.
And that's a mini graph cactus-based graph.
Yes.
For the variant calling. Yeah, and I'm just thinking, what do you think that maybe seeing that a lot of these disorders are sort of in these more complex repetitive regions, that the structure variants identified when you build the graph through PGGB, which looks at a lot of more of these complex regions, do you think that those might somehow be linked to diseases and that we overlooking them in the MC graphs?
I think it's definitely possible. One thing that we sort of have grappled with is our unsolved, when we first started this, we were not sure what we were going to find. And it was interesting that our unsolved rate is quite comparable to international report, from other groups. And so it suggests to me that there's something that we're all missing, right? This is not just an ancestry, some peculiarity in our setting, because we're studying genomes that are underrepresented. So there's some sort of central thing that we all not getting at. So it's either that's why my money's on the structural variants. I think that's where that's going to be very important, and probably these repetitive regions of the genome, which all of us haven't fully examined yet.
Yeah, I guess that'll be the interesting thing, once we have enough genomes to be able to make the comparison. We build a graph and be able to interrogate these sort of structural variants in the general population and understand those a bit more. Then I think that's the sort of first step. But it's cool. And the other thing is it could be just another mechanism that we're not looking at, and made me think about the whole methylation aspect that you get out of long read data as well now, which allows us to interrogate that as well. So lots of.
I think also just to go back to Chris's comment about the coverage, that is also something we thought about with the Revio. It's obviously it can be made cheaper if you accept lower than 30x. But I also, wanted the ability to do the assemblies and also, contribute to the RefGraph project, at least for the first phase. So that was kind of the decision around sticking with the 30x per sample initially.
Great, thanks.
Okay, I don't see any more questions in the chat box or any hands raised, but I see Chris has just raised his hand. Chris, you can go ahead.
Sorry.
No worries.
It's actually just more of a follow-up question. It's sort of a general one, because when we did the workshops, you kind of jumped in feet first, I would say, and really made a lot of really interesting findings with the data that you have, and were able to apply it. So you may have a perspective that's not that we may not have, because we have different aspect of it. But what would you consider to be an impediment to somebody that's interested in using graph-based techniques for genome analysis? I mean, what do you think they would need to know going in? What kind of knowledge do they need to come in with in order to really take full advantage of these newer techniques?
Sure. That's quite I think for most people, it would feel quite daunting. Any move to a different way of doing things, like a different analysis pipeline, is obviously a big ask. And I think part of this preliminary, work that I've been doing is trying to see what value, what advantage does it offer in a really practical sense for a particular application, to kind of see if that's if something that other groups or other users should consider. Like, where does it really offer you an advantage over using a linear reference? But I guess, I don't know a lot of people, particularly in the clinical space, who are using NGS in a kind of clinical translational way. So I suppose it might be a long time before we see the adoption of pangenomes in this space, I think.
Yeah, that was my impression here as well. It's going to take some time to show that it's showing advantages.
There are even some consortia that I work with that send stuff on HG19. Not infrequently. So I guess it might be challenging.
I definitely think in the research area there's obvious interest. But I think the clinical space is going to be the hard one to crack. But I think it'll.
And I guess I have a unique setup in that this is being done in a research setting, not in a clinical diagnostic setting. So although we try to push the translational agenda and we give feedback to patients, we obviously have a lot more freedom, exploratory freedom, to kind of push the boundaries and try new things and try to be at the cutting edge. Whereas, workflows that are confined in a much more rigid clinical diagnostic space probably don't have that flexibility and have to stick with tried and trusted ways of doing things.
Yeah. Well, thanks.
Thank you for that. It's been some great engagement. I don't see any questions or hands raised from the participants. So I see Sean, you've got your hand raised. You can go ahead.
Sorry, we're going to carry on interrupting you. Yeah, no, I just wanted to see. I don't know, this is just a bit of a maybe not the best thing to ask, but if there's anyone on the call, I mean, we're running these seminars as sort of a build-up to a workshop that we're planning. So I don't know, since we've got some time, is there anyone on this call who's got a keen interest in, making use of an African-based genome graph, just so we can get a gauge of who the people are? If you're keen to let us know, you can feel free to unmute and let us know if there is anyone on the call. I expected it to be quiet. But yeah, I just thought I'll try and see if anyone. So we know that people are interested, and people that do apply for the course, we can make sure that we can assist them. But that's fine. That's fine. I think we'll be able to gauge that from the applications. Thanks, Nicole.
Thanks, Sean. And just to follow up on that, it might be a good time to let people know that we will be hosting a workshop in October, which is on human pangenome bring your own data analysis workshop. So I think maybe after this session, if you find that it's relevant to your work or you want to explore more on it, you can visit the workshop page, which I will share here. It is a competitive application or selection process. So you are welcome to have a look at the workshop announcement and see if it's relevant for you and submit an application. If you require more information, you can contact either myself or Sean or Samir, who's also on the call, to find out any other information that you might be interested in. And to follow on to that, we will have another webinar taking place on Friday, the 13th of September, starting at 4 p.m. CAT. We will have a talk from Andrea Guaracino and Flavia Villani. They'll be presenting on two different topics. Andrea will be presenting on pangenome-based structure deconvolation of the amylase locus, while Flavia will be presenting on enhancing complex trait mapping in recombinant inbred rodent strains through pangenome analysis. So if you've registered already, you would have seen some of those talks that will be happening on Friday. So yeah, I invite you to actually visit the website. I've sent it to one person in the group. I didn't realize it. I'll just post in the chat again. That's the link to follow for any other information that you want on this. And yeah, you can either contact myself or visit the website, the webpage, and submit your application. I will say that on top of that, I would like to thank Melissa for your presentation. It was very informative, and the participants were very engaged in what you had to say, which shows that there is interest in this. And as Goodwin is saying, it's very informative and insightful. And with that, yeah, I'd like to thank you. Thank you, Melissa.